Hello student, I hope all of you are fine and today we are going to discuss about the biotechnology and its chapter the culturing of the microorganism. So in the last lecture we have discussed about the culturing process of microorganism, how to culture the microorganism in the laboratory condition and what is the requirements. We are studying about the media, which type of media are favorable for growing the different microorganism and the producing the commercial products. So in the continuation of that, and what is the procedure, you are choosing the media, आपने media choose कर लिया, after that what to do, we are going to sterilize the media. Yeah, so it's a very important step, the media sterilization, because if you are not sterilizing the media properly, so the unwanted growth of bacteria take place or unwanted growth of microbes is take place, यानी वो microbes जो आपको requirement नहीं है वो grow हो जाएंगे. Clear? So that's why we are going to sterilize the media very well. Clear? So in this topic we are going to discuss how to sterilize media in the laboratory condition and how to sterilize media in a industry condition. Clear? So how to sterilize media in industry. So for that, if we are working in laboratory, so you are taking the media in small amounts. It's 100 to 1000 ml media is taking in the flask and sterilize this media in the autoclave. Clear? So we are going to sterilize this media in the lab conditions in the autoclave. So we are keeping the pressure, it's 15 psi and for the time it's 15 to 20 minutes and it's a temperature is 120 degrees C. Yeah. So this practical you have already performed in the 11th class in the biotech lab. You are preparing the media preparation. During the media preparation, you have prepared media. After that, you sterilize the media in the autoclave. So it's the same thing. Yeah. So that is written. It's the same thing. You take the media and you prepare the media. After that, dissolve this media in the water. Then keep the cotton plug on the flask and after that you keep the media in the autoclave for the sterilizing. So this is the lab condition. But if you are working in the industry, so how to sterilize the media in industry? So in the industry we can use the huge amount of the media because we are going to produce the product in the large scale. So we are taking the media in large amount. Yeah. So this media can sterilize in outside or fermentation. Clear? Or in the fermenter. So what is fermenter? In the next topic we are going to discuss about the fermenter. Fermenter is also called the bioreactor. Clear? So fermenter or bioreactor is a vessel in which we are using the media for the sterilization, as well as this vessel is used for culturing microorganisms in the laboratory. In the industry. So for the industry point of view, steam is used for the sterilization of the media that is used during the fermentation process. So because this media is used during the fermentation process, so this media is also called the fermentation media. So it's a process, that is how to sterilize media in the industry and how to sterilize media in a lab condition. So that is one jacket is present in the bioreactor in which we pass the stream, and after that the other jacket where the cooling water is passed. So these are the process or in the sterilizing the media in the bioreactor. So as you know, steam is passed and this steam is heated for 15 psi and for the 20 minutes for the proper sterilization of the media. And next thing that is important is aeration. Clear? Aeration is a very important. So first you take the media, sterilize the media. After that, if your microbes is a aerobic microbes, so the proper aeration is required for the growing of microorganism. That's why in the lab conditions we are using the equipment. It's a shaker. Here in lab conditions we are using a very simple instruments. You can see in this figure, and here is a holding is present here. Holding stand is here in which we are keeping the flask. You know what is the flask? You are using this flask in your biotech lab in the APS. So that's why we are keeping the flask in this holder, and then hold these flask and then start, and it's a shaking. Yeah, so just shaking the flask so that aeration is proper take place in the flask. So this situation are used in the lab condition. But if you are working in the industry, so in the industry the aeration process is inbuilt in the bioreactor. Yeah, bioreactor is a vessel in which you are sterilize the media and you are growing a microorganism in this uh, vessel. So that's why the aeration process is properly maintained in the bioreactor. So for that, the stirrer is used for the mixing the media in the bioreactor. So first thing, you are taking the media and then sterilizing the media properly, and we are studying how to sterilize media in lab condition and how we sterilize the media in the industry point of view, and then same here how to maintain the aeration or mixing of oxygen. Yeah. So in the lab condition by using the shaker, and in the industrial point of view we are using the bioreactor. In the bioreactor there is a other part that is stirrer that is used for the mixing of oxygen or mixing of uh, different substances also in the medium properly. So that is used. And baffles, what is baffles? We are going to discuss in the next lecture, and when we studying about the structure of bioreactor and functioning of bioreactors also. So some basic equipment that is used during the culturing the microorganisms, equipment for microbial culture. It's a baffle flask. You can see this is a flask. So the mouth of this flask is like this. Some producing end is present that is for the proper culturing of the microorganisms and increase the efficiency of oxygen transfer. Yeah. So this is a vessel that is used in the lab conditions for the growing the microorganism, and it's a shaker that is used for the mixing the oxygen in the media. And next we are going to discuss about the bioreactor. So what is bioreactor? Yeah. So in the next lecture we are properly discussing about the bioreactor, and this is the incubator shaker. You can use this type of shaker also, and this advanced shaker is also coming. So it's also working as an incubation. Incubation means this type of shaker is maintaining the temperature also. Yeah, if you are wants to a proper temperature for the growing microorganism, so that this chamber is used that maintain the temperature also. So I think all of you are understand, and thank you for watching this video. And if you feel any problem, then tell me.